Hello everyone. Welcome to the new session of cell cycle and cell division. So this is the first part of this chapter. So what is cell cycle? Cell division is a very important process in all living organism, right? So this is the phase in which cell divide, right? Cell divide. So this is a very important process in all living organism, right? Because living organism, when it divide its cells, one cell divide into two, okay? So the continuity of life depends upon the cell division. If we talk about the gametes, formation of gametes also, that also required the cell division, right? So this is a very important process in all living organisms. During the division of a cell, DNA replication and cell growth take place. So this is very important. The nuclear content, suppose this is a cell, it is having a nucleus, and inside the nucleus DNA is present in the form of chromosomes. So this chromosomes undergo DNA replication, means the DNA makes copy of it. So that is also called as duplication, right? So now here we can see the nuclear content of the cell becomes double. It's become double, right? So what happened? The nuclear content is divided into two nucleus in one cell. Okay, so this is the two nucleus, and later what happened? The cytoplasm also divides and form two daughter cells like this, right? Two daughter cells. But this process is not so simple. As I have just shown here, this is not so simple. It is a very complex process, right? So the during of the cell, DNA replication and cell growth take place. So how this occur? In which phase of the cell cycle it occur? We are going to study all this in this particular session. Okay. So cell division, DNA replication and cell growth take place in a coordinated way to ensure the correct division and the formation of progeny cells containing intact genome. So what are the progeny cells? Progeny cells means new cells formed from the pre-existing cells. So that is called as the progeny cells, right? So one cell, okay, it's having a nucleus. So first of all, its nucleus divides into two, and later on the cytoplasm also divide, and it's having two daughter cells. So this is the karyokinesis, that is the division of the nucleus, and this is cytokinesis, that is the division of the cytoplasm or the cell, right? So here these two are called as progeny cells. So this I've already told in the previous slide that when the cell has to divide, replication process take place and the DNA content of the cells gets doubled, right? So here the nucleus, which is having the two nucleus here in a cell, right? So it is having the same nucleus. It is having same DNA. It is having this, both are having the same DNA. So that division, the karyokinesis has to be in a very coordinated way, to get ensures that the correct division is taking place and the formation of progeny cell is containing the intact genome. It is having the same genome from which this cell is formed, right? Both these progeny cells is having the same genome from which the cell has been formed, right? The sequence of events by which the cell duplicates its genome, synthesizes the other constituents of the cell, and eventually divides into two daughter cells is termed as cell cycle. So this is the definition of the cell cycle. What does it say? The sequence of events. So there are many events in which the cell duplicate itself, right, or divide itself. So that cell cycle, that is the coordinated stepwise things occurring in a manner and in which the cell duplicates its genome, the DNA gets doubled, right? That cell duplicates its genome and synthesizes the other constituent of the cell also, like many enzymes and many proteins are required for this genome formation, for the duplication of this genome. So that synthesis also takes place in this cell cycle, right? And eventually divides into two daughter cell, and that is called as cell cycle. Okay. Although the cell growth in terms of cytoplasm increasing, it means that the cell increases its size. The cell increases its size. Increases its size. That is called as growth of the cell. Right? So when the cytoplasm increases, okay, the size of the cell is there. The size of the cell increases, right? So that is called as the growth of the cell. Is a continuous process. So this cell growth is a continuous process in which DNA synthesis occur only during one specific stage of the cell cycle, right? So the cytoplasmic increase, it called as growth, right? But there is only one specific stage in which this DNA synthesis occurs. So this is a very important step in the cell cycle. The replicated chromosome DNA are then distributed to the daughter nuclei by a complex series of events during the cell division. So this, in the case of cell cycle, we are having one phase that is also called as the cell division, in which the daughter nuclei is distributed, okay, in two different cells. And this is not a very simple process. It is a complex series of events. It is a complex series of events. So what are the phases of the cell cycle? These events are themselves under the genetic control. Okay, so this cell cycle is itself in a control of genetics. That is called as genetic control, right? So a typical eukaryotic cell cycle is illustrated by the human cell in culture. So these cells divide once in approximately every 24 hours. So here we are going to study about taking an example of a eukaryotic cell, right? That divides in every 24 hours. So if you take a human cell in a culture form, in a laboratory, then you can see that the cell is dividing in every 24 hours. So here comes the different phases of the cell cycle. Duration of the cell cycle vary from organism to organism and also from cell type to cell type, right? So organism to organism, this G1, S, G2 and M phase. These are the different phases of the cell cycle, and it varies from organism to organism. Time varies from organism to organism, and also in an organism, cell type to other cell type, right? So yeast, for example, can progress through the cell cycle in only about 90 minutes. So in 90 minutes, the yeast cell can undergo or complete it one cell cycle. Okay. So there are different phases of the cell cycle. The cell cycle is divided into two basic phases. The first one is interphase and M phase, that is the mitosis phase, right? So M phase, here we can see it is divided into prophase, metaphase, anaphase, telophase, and the cytokinesis part. So that is the prophase, metaphase, anaphase, and telophase. These four part is called as karyokinesis. So karyokinesis occur in these four different steps. And there is a cell division of the cytoplasm, division of the cytoplasm by forming a membrane around it, and that is called as the cytokinesis. Now, what is an interphase? So here we are having G1, S and G2 phase. This particular part is called as interphase. So now you can see this interphase is the major part of the cell cycle. It is a major part of the cell cycle, right? So M phase represent the phase when the actual cell division or the mitosis occur, and the interphase represent the phase between the two successive M phases, right? So M phase, what happens in M phase? Actually, the actual division of the cell takes place. That is called as cell division. So that cell division can be divided into two steps. That is karyokinesis and cytokinesis. Now this karyokinesis, that is the division of the nucleus. Right? And cytokinesis is the division of the cytoplasm and forming the membrane bound. So it is significant to note that in 24 hours average duration of the cell cycle of a human cell when it is cultured, and cell division proper lasts for only about an hour. Okay, so cell division occur in only 1 hour, but the total cell cycle which constitute interphase and M phase, this is called the cell cycle, and this is for 24 hours. But if we talk about this cell division, it takes place just an hour, that is in 1 hour. So 1 hour karyokinesis and cytokinesis division occur. But what about the 23 hours? This more than 95% of the cell cycle is of interphase. That is the G1, S and G2 phase. That is the major part of the cell cycle, right? So interphase lasts more than 95% of the duration of the cell cycle. So this is very, very important. M phase start with the nuclear division corresponding to the separation of the daughter's chromosome, that is the karyokinesis. So separation of the daughter chromosome, that is called as the karyokinesis, means the division of the nucleus, and usually end with the division of the cytoplasm, that is called as the cytokinesis. So this occur in the M phase. This is the M phase function or the M phase significance, right? So karyokinesis and the cytokinesis part that occur in the M phase, right? The interphase, though called the resting phase, is the time during which the cell is preparing for the division by undergoing the cell growth and DNA replication in orderly manner. So this interphase, that is also called as the resting phase, but this is the most active phase of the cell cycle, right? So this is the time during which the cell is preparing for the division. Okay, so cell is preparing for the division, okay, by both cell growth and DNA replication. So DNA is duplicating itself, making its own copy, okay, by the process of replication, So this is a very complex process which is occurring in interphase. So interphase is actually not a resting phase, but we cannot see such type of changes as we can see in the M phase, right? You can see M phase having division of the nucleus, movement of the chromosome. Okay, so that you can see. So it shows that this is a very active phase, right? But such type of things cannot be seen in the interphase, but actually in this resting phase or the interphase, major changes is going in the nucleus. So here we are talking the daughter chromosomes are dividing. So how the daughter chromosome are dividing? Because the DNA has been doubled. The DNA content has been doubled. And what happened? Then that DNA content which has been doubled is separated in the process of karyokinesis. So now you can understand that this DNA replication is how much important. Without this, there will be a no karyokinesis. So actually, the interphase is not a resting phase. It is one of the most active phase of the cell cycle, right? Okay, so the interphase is divided into three further phases. That is the G1 phase, that is also called as gap 1 phase, S phase, that is the synthesis phase, G2 phase, that is also called as the gap 2 phase. So these three phases are the part of the interphase. Okay, the interphase can be divided into these three parts, that is the G1, S and G2 phase. Okay, so we will see one by one that what changes occur during these phases of the interphase. The first one is that is the G1 phase. So G1 phase corresponds to the interval between the mitosis and the initiation of the DNA replication, that is the S phase. That is the S phase. So this G phase is the G1 phase is present between the M phase and S phase or the synthetic phase, right? It is present in between these two phases. During the G1 phases, the cell is metabolically active and continuously grows but does not replicate its DNA. So whatever the things is required for this S phase, that synthesis occur in the G1 phase. So in S phase, what happened? DNA replication takes place. DNA replication takes place. So whatever the enzyme, that is the DNA polymerase, right? Gyrase, ligase, they're different enzyme. So all that gets synthesized in this G1 phase. So cell is preparing itself to get enter to the S phase, and for that they require many types of enzymes and proteins. So all that things get synthesized in the G1 phase. So that is why it is called as metabolically active phase, right? The G1 phase is the metabolically active phase. Okay, so next phase is the S phase. This is a very important phase of the interphase. Okay, so S or the synthesis phase marks the period during which the DNA synthesis or replication takes place. So this is the phase in which DNA replication or the DNA duplication, doubling, okay, doubling of the chromosomes or the DNA content is occur. So during this time, the amount of DNA per cell gets double. So if we see on this graph, right? So G1 phase, it is having, suppose it is having the X content of the DNA. So it is same from the starting of the G1 phase and at the end of the G1 phase, it is same. The DNA content is not increasing, right? Now, what happened in the S phase? So here you can see DNA replication is taking place in this particular region. DNA replication. That is copying of its own DNA, right? So if the DNA is in the 1x, now after the completion of this S phase, it will have 2x. It will have 2x. So if the initial amount of the DNA is denoted as 1x, then it increases to the 2x at the end of S phase, at the end of S phase, right? So during this time, the amount of DNA per cell gets doubled. Okay. There is no increase in the chromosome number. The chromosome number is not increasing. If the cell had a diploid or the 2n number of the chromosome, okay, diploid means every chromosome is present in the paired form. Every gene is present, which is present on the chromosome, are in the paired form. So that is called as 2n number of the chromosome at G1 phase. Even after the S phase, the number of the chromosome remains the same. Okay, it is same. In animal cells, during the S phase, the DNA replication begins in the nucleus and the centriole duplicates in the cytoplasm. So in the animal cell, in the nucleus, replication of the DNA taking place inside the nucleus, and what is occurring outside the nucleus, that is in the cytoplasm, centriole duplication takes place. So very interesting. In the animal cell, inside the nucleus, DNA duplication taking place, and outside the nucleus, that is in the cytoplasm, centriole duplication taking place. Okay. Okay, so we talk about the S phase. Now comes the G2 phase. During the G2 phase, the protein are synthesized in the preparation for the mitosis. Now, there was G1 phase, there was S phase, there was G2 phase, and then comes the M phase. Now, you saw that in the G1 phase, what happened? It synthesized the enzyme which is required for the S phase, right? After S phase is completed, in this S phase, major thing, that is the DNA replication takes place, right? DNA replication takes place. Now, what happened in this G2 phase? M phase required lots and lots of enzymes, So these enzymes, which is required for the M phase, get synthesized in the G2 phase. As we have seen in the case of G1 phase, whatever the enzyme, whatever the proteins required for the S phase get synthesized in the G1 phase. So here also, whatever the enzyme and proteins is required for the M phase gets synthesized for in the G2 phase. It means that the cell is preparing itself to get to the M phase, right? So during the G2 phase, the proteins are synthesized in the preparation for the mitosis while cell growth continues. So some cells in the adult animal do not appear to exhibit the division like heart cell. They never undergo the division. Okay. Heart cells never undergo to the division, and many other cells divide only occasionally as needed to replace cells that have been lost because of the injury or the cell death. So there are many cells that divide occasionally, sometime only, and that too when there is a injury or cell death. But some cells never divide, like heart cells. So these cells that do not divide further exit from the G1 phase. They get out from the G1 phase to enter an inactive stage or the quiescent stage, that is also called as G0 phase of the cell cycle. So here you can see G0 phase. The cell which never divide goes from G1 to G0 phase. G1 to G0 phase. Very, very important point, right? Cell in this stage remain metabolically active but no longer proliferate unless called on to do so, depending on the requirement of the organism. So when required, the G0 phase can jump into G1 phase, right? So in animals, mitotic cell division is the only seen in the diploid somatic cells. Okay, so we are having two types of cell: somatic cells and germinal cells. Germinal cells or germ cells. Germ cells. So where these germ cells are present? These germ cells are present in reproductive organ. And these reproductive organ having a special type of division that is called as meiosis, right? And that give rise to haploid gametes. Whereas the somatic cell, so we are having two types of cells, somatic cell and germinal cells, right? Now somatic cells, that is body cells. So body cells are formed by mitotic division. So there is M phase, right? This M phase can be mitotic or meiosis. So if it is a somatic cell, then it will undergo simply mitotic division. And if it is a germinal cells or the germ cell, and that cell is only present in the reproductive organs, right? And that undergoes the meiosis division. So there are two types of division, mitosis and meiosis, right? So in animal, mitotic division is only seen in the diploid somatic cell. So I told you just because this term somatic cells came here. So now you can understand what is somatic cell. That is means the diploid body cell, simple diploid body cells. Okay. So however, there are few exceptions to this where the haploid cell divide by the mitosis. For example, male honeybee. There are some examples like the haploid cells divide by mitosis. Haploid cell undergo mitosis. Okay, so these haploid cells undergo mitosis, right, and form male honey bee. So this is an exception. Against this, the plants can show mitotic division in both haploid and diploid cells, right? So Chlamydomonas is one of the example of this. Alternation of generation in plants identify plant species and stages at which mitosis is seen in the haploid cell. So generally mitosis is occur in the haploid cell. So haploid cell means single set of chromosome, n number. So from n the cell divide and it again form n. This cell is again divide and it form n. So this is mitosis division, right? So this we will see in another session, that how does the mitosis and meiosis divisions takes place. M phase. Now here comes the M phase. This is the most dramatic period of the cell cycle, involving a major reorganization of virtually all components of the cells. So here we can see a major changes and reorganization of the cell component, whether it is present inside the nucleus or in the cytoplasm. So since the number of chromosome in the parent and the progeny cells is the same, it's also called as the equational division. So we will see all this in one particular session. Though a convenience, mitosis has been divided into 4 stages of the nuclear division, that is the karyokinesis. It is very essential to understand that cell division is a progressive process and a very clear-cut line cannot be drawn within between the various stages. So you cannot intermix the different stages. The stages which you have studied today, that is the interphase and the M phase. We cannot overlap the phases, right? The G1 phase cannot overlap with the S phase, or S cannot. There is a clear cut demarcation that the cell is in G1 phase, the cell is in S phase, and the G2 phase that completes the interphase. And later comes the M phase. Now M phase, it is having a clear cut karyokinesis part, that is the prophase, metaphase, anaphase, and telophase type of division, right? And they are having a specific movement of things or specific changes of things. So that is fixed, that this particular change occur in prophase, this particular change occur in metaphase, right? So this is a very clear cut line and cannot be drawn between the various stages. Okay, so there is no overlapping of the program or the overlapping of the stages in between, right? So the M phase can be studied in the following steps. Karyokinesis involve the 4 stages, that is the prophase, metaphase, anaphase, and the telophase. So this is the part of the karyokinesis, right? So this is all about the cell cycle, right? So cell cycle is what? Cell cycle constitute the 2 very important stages, that is the interphase and the M phase. Now generally we see that the M phase is very important, but the cell prepare itself for the M phase undergoing interphase, when it undergo interphase. So in interphase there is a 3 very important steps, that is G1, S, and G2 phase. Okay, so we have seen what all the changes that takes place in these interphase divisions. Okay, so this is all about the cell cycle. We will meet with one more session for discussing mitosis and meiosis. Till then, thank you very much.